So now let's talk about how to view the quality of each of your individual reads. First, let's talk about what high quality sequence looks like. Here you can see 4 electropherograms stacked in order of quality from high down to low. Each little wave in these images represents the fluorescent signal from an individual ddNTP nucleotide as it runs through the gel in a Sanger sequencer. The color of each wave represents a different nucleotide, A, T, G, or C, which ultimately gets translated into your sequence. Hopefully it's clear that the waves are clean and clear in the high quality sequence at the top and become increasingly ugly and garbled and uninterpretable as you move down to the horrible sequence at the bottom. From there you can go to the upload and drop down and you can see all of the samples that you imported. Each of these sample names represents a sequence, and you can see some of them might have a little warning with an exclamation point. We'll talk about that in a bit. If you do the toggle, you can click View Sequence, and then you can view your sequence. Typically, when you look at a sequence, you're almost always on the front end going to have a bunch of ends, which represent bad calls or bad sequence, and you'll see the same thing at the back end of your sequence, a bunch of ends at the end. But typically, all the stuff in between those two front and back tails look good. If you click on AB1 view, you can see the normal sequence view or the electropherogram. Running along the top will be the individual base calls. In the middle, you'll see a quality score for each of those base calls. The blue bars represent the quality for each individual call, and the small blue line running down the middle represents a threshold, what's called Phred score. You can also see the position of each of those base calls or account for each of those base calls. And you can scroll across and you can see that at the far ends, there's typically a bunch of ends with very low bars or Phred scores, not surpassing the threshold for high quality. Whereas in the middle, you see most of those calls have clean and high Phred scores. Now let's go down and take a look at Louisa's reverse read that has that warning sign that indicates it's a lower quality sequence. Here you can see, similar as before, there's a bunch of ends at the front end and back end, but there's also a lot of ends that we're seeing in the middle of the sequence. This is typically indicative of a lower quality read. Let's take a look at the AB1 view, and what you'll see is the waves at the sequence at the bottom are all garbled, overlapping each other and not clean. And the individual purple bars or Phred scores for each of those calls rarely pass that Phred score of 20 threshold. And so most of these calls are low quality calls that can't necessarily be trusted. Question: how many of the 8 practice reads are low quality? Please hit pause until you know the answer. So the answer is 1. Only Louisa's reverse read. That's the only practice read that has one of those exclamation marks. Next question: what exactly does it mean if a read has the low quality exclamation mark next to it? Please hit pause until you know the answer. A low quality score alert means that the average error rate for this particular sequence is greater than 1%. This indicates that the sequence is of low quality and may produce erroneous analysis results. So in other words, the sequence is less than 99% accurate. Technically, it's still okay to analyze these sequences further downstream if the software enables you to, but just take those interpretations and analyses with a grain of salt because they may be producing and are likely producing erroneous results. A quick review of Phred scores or Phred quality scores. Recall that these are the probability of an individual base call in a DNA sequence being correct. The Phred scores are indicated here by these lines and bars. Now the blue line represents a Phred score of 20. And if an individual bar exceeds that Phred score of 20, it means that that individual base call has a probability of greater than 99% of being correct. And typically a Phred score of 20 is the threshold for an individual base call to be considered of good quality or 99% accurate. So once again, if gray or purple bars in your electropherogram of your sequence pass above this blue line, it means they have a high enough Phred score or are accurate enough to be trusted. So given the information from the previous slide, which base call in the image above is most accurate? Base call A, B, or C? Please hit pause until you know the answer. If you said C, you're correct. C is the most accurate. It has a Phred quality score above 20 and therefore has a greater than 99% probability of being correct. Both A and B have Phred scores just below that threshold. Next question. How long is Jessica's reverse read? Please hit pause until you know the answer. Hopefully, you were able to figure out that you could toggle to the end of her read using this little bar here on the left and scrolling all the way over to the right. And you should have seen that the read ends at about 670 base pairs, as you see in the red arrow on the right.